My name is Michael Gilpin. I am a fifth-year undergraduate at the University of Wisconsin-Madison. I'm working with Daniel Hayden. He's a grad student at the University of Wisconsin-Madison. We work in the plant pathology department. And I'll share my screen. Okay, there we go. So my project was using microbiomes to understanding indigenous agriculture. The project is modeled after the way indigenous agriculture is. So modern day agriculture is really monocultures, and these are just fields where they have one crop planted across the entire field. And this way of agriculture is really it lacks diversity, and indigenous people early on for thousands of years have recognized that a highly diverse ecosystem do better, and they try to imitate the natural systems compared to having just like a monoculture field.
Michael, can I interrupt you for just 1 second? Would you mind putting it on presentation mode on your PowerPoint so it's a little bit small to read? Just click it on the little.
Oh yeah, okay. Oh. Here we go.
Awesome, thank you. Miigwech.
All right. Yeah, so I was saying this way of agriculture has been done for thousands of years. It's highly diverse, and this way of agriculture does better. A lot of the ways are deemed sustainable and organic have actually been implemented for thousands of years. A new way we are beginning to understand this natural system is through microbes. Microbes play a major role in our environment. They mediate processes like nutrient cycling and diseases. The reason why you see certain plants in one area compared to another, or why you don't see one plant dominate an entire area, is because of these diseases. These diseases help manage and limit the distribution of these plants. Basically, we want to understand what microbes are there and what they do may help us understand how these natural ecosystems function. So basically, we just want to understand what are all the microbes there and what is the relationship and what is going on. So kind of how we do this is we take sample roots from the soil, from the field, and we break them down. We extract the DNA from these roots, and kind of the more technical way of how we do this is we look at the DNA genes, specifically 16S and ITS, and we look at these two genes because this is the ribosomal RNA in all organisms, so that's how we pull out the bacteria and fungi sequences. Then we take all of these sequences and we run through a bunch of code to pull out all of the names of the organisms of what is there, and then from this large data set of thousands of entries of organism names, we're able to run statistical tests in R to understand what is really going on, what are the differences, and look at the relationships. We have a collaboration with Iowa State University. We're working with a indigenous grad student there. He's looking at the ancient garden sites, and Three Sisters project is looking at kind of the land and the garden that was used by the Meskwaki and Potawatomi nations. Basically, we're looking at if there's differences in chemical and microbial composition of like the garden soil compared to nearby soil that isn't being used. And basically, we're just trying to look at the differences between the chemical and microbial compositions. To give an update, we have completed the sequencing of DNA of soil samples. So we ran, we extracted all of the DNA, and we did the analysis on thousands of sequences in QIIME. We now have to do statistical tests to link the soil chemical composition to the microbes. So now we're at the point where basically just going to do all of the tests in R to analyze all of the data. We worked with many different tribal growers in Wisconsin to implement organic practices. We work with Dan Cornelius, work with a Oneida White Corn group, a Whole Chunk Organic Farming group, and a Menominee nonprofit. We're trying to implement organic practices. Like I said, we're taking their, so all these sites have corn. So we're planting the rows of corn, and then to promote diversity in between the rows of corn, we're planting cover crops, and these cover crops, help promote diversity. Usually, like with modern day agriculture, people will just, harvest the corn and then get rid of the corn and then plant the cover crops in place of the corn until next harvesting season when they plant the corn and do that process over again. But we're planting the cover crops in between the corn to promote diversity. So this was at the Ho Chunk field. Here's me. I was planting some corn. Conclusions and future. I'll help Daniel do the DNA sequencing and microbial analysis on the corn roots from all of the sites and wrap up our Iowa State University collaboration. That's where we're at now. That concludes my presentation.